thank you very much. As said, I am Paul Agapow from AstraZeneca, and I am the stupidest person in this room. But I do have a certain animal cunning that's going to come into this talk. So, as said, once upon a time, I was a lab coat-wearing biochemist and immunologist. Along the way, computers happened to me. I became that amorphous title, a bioinformatician, that even more amorphous title, the computer guy, hanging around university labs and so forth. But I am now a health informatics director at AstraZeneca in the delightfully titled Oncology R&D Real-World Evidence. And by the end of this talk, those words will make a little bit more sense. As a performer, no conflicts of interest or anything like that. Now, do be very clear. This is certainly a work in progress. Things did not move as fast as expected. There were obstacles, of course, that surprised to us. And part of my talk is going to be about those lessons to all of you to make you live a better life. Now, we are certainly not experts in the world of graphs and graph theory, but it is a subject that is emerging within pharma. People are getting very interested in it, and this is a first step in it for us to develop expertise, to explore, to see where we can go, okay? So we are people with problems to solve, okay? Now, we have made lots of mistakes. A final useful point is a lot of the data that's in here we are very tricky about the sensitivity of. Nothing you could photograph, take away, and sell for money, but therefore I've obscured a lot of examples and just put in synthetic ones in there to give the idea of what's going on. So, the pharmaceutical industry and data science. I might add that I was in universities till about 14 months ago or so I have that eye of the outsider on pharma and its foibles and its differences from the university. But as said, my pitch is what we call real-world evidence. Real-world evidence is one of those big trending words out in the world there, sometimes real-world data, sometimes real-world analytics, okay? And it basically means messy data. Anything that's not obtained from within a rigorously controlled clinical trial, anything that's not a table. We're talking about electronic health records, we're talking about social media, fitness trackers, claims registries, databases, paperwork, anything we can pull a signal out of. So one of the reasons, well, there are several reasons pharma is interested in this. One is that it's cheap. Number two is that it gives you access to data that you would not get any other way. You know, it covers a number of people that you couldn't do in an experiment. It covers a time span that you couldn't access at. It involves questions you could not pose upon a population because of ethical considerations. That's what I do. It's also more realistic. It's a signal of the world. If you have a look at experimental populations and clinical trial populations, they have one predominating characteristic, and that is that they are not typical. Drugs tend to get tested on white, young, male university students, or if you're moving to the sick population, we're talking about people with uncomplicated diseases, populations that are somehow pure. Well, that's a little bit of a sidestep. Let's talk on to our actual problem. And this is to tell you why it's so hard to develop drugs. Of course, this is a big point in our world, the cost of drugs, how expensive and slow it is to visit drugs, and there are many good reasons for this, okay? It is a hugely protracted and difficult procedure. For a start, you've got to find yourself something in the subject, something in a human that possibly you could get a molecule or a drug to interact with. And then you start with a whole bunch of candidate molecules. Typically, say, for instance, you might start with, say, tens of thousands. Pharmaceutical companies have all of these on their books. Then by the time you winnow those down to those who can be delivered in a reasonable way, to those that can be manufactured reasonably, you're probably down to around about 250 before you've even gotten to the point of putting it inside a human. Then we move to the clinical trial phase, which is what I'll be talking about chiefly today. And by the time you go through the clinical trials and test it for safety and efficacy and so forth, by the time you go through all of that, at the end you may come out with one. 10,000 possible candidates down to one, and in between we have $2 billion and about 10 years. This is called the valley of death, okay? Developing drugs is like going into a casino, slapping 100 million down and saying, bet it on the zero. That's the sort of world we live in. It's very expensive. It's only for deep-pocketed players. It's very random, okay? When you're paying for a drug, you're not paying, in a sense, for that drug being developed. You're paying for all the failures. So what can we do about the failures? Now, as I said, clinical trials is kind of where I'm going to be coming at for this. Clinical trials is a very rigorously regulated and tight device. It's a very strict and formal thing, and it goes in these three phases: phase 1, phase 2, and phase 3. And in phase 1, they ask the question, does this drug accidentally kill you? Phase 2, they say, seriously, does this drug actually kill you? And does it do anything good? And in phase 3, they say, seriously, does it kill you? Does it do any good? And does it do good better than any of the competing things? First phase tends to be tested on around about tens of people, the second on hundreds of people, the third on thousands of people. And each of those patients in there will cost upwards from $10,000 each. It's an expensive process. Now, the more subjects you have in a phase, the more statistical power you get out of that, which is important when you're talking about the messy complexities of the human body. And you'll end up with all these curves like this on the right there, where people say, well, the tumors were 20% smaller in the treated patients than they were in the controls, so we think that's arguable. But the sad truth of the fact is that 60% of trials never get the number of patients that they want. And this makes things hard for us, okay? You've got a more statistically challenging subject there. How can you complete your trial when you haven't made your targets? So forth. And this is sort of the central problem we're looking at today. So in the old-fashioned days, we would get patients for trials in a social grease sort of way. People would know people. They would reach out to contacts at hospitals. You would put up advertisements around the place, send out brochures. Okay, you would even have sites for volunteers. Clinical research organizations would advertise for people willing to live in a room for 28 days and take their drugs and eat their food. In our brand new modern world, that's not going to work. We're putting many, many more drugs through the pipeline now. Okay? But fortunately, we have, to some extent, these new things, federated EHRs, electronic health record systems. There are these major providers like Trinetics, pictured there on the right, where they promise to say, if you tap into this, okay, you get access to 120 million patients in the United States and 100 million patients in Europe. However, it would immediately occur to you, of course, there is more than 120 million patients in the United States, and there's more than 100 million in Europe. There's this whole body of patients out there that are not findable, okay? Their records, as they are, even these ones that are in the system, they may not have sufficient detail for us because we have quite discerning lists of criteria for the patients we need to find. And pharma also has preferences for how it likes to recruit people to talk about. So, typically, for a trial, you might have a set of what we call inclusion-exclusion criteria, which can look quite exotic. Postmenopausal women in Spain, okay, they have not used platinum therapy, they have this particular biomarker. It cannot be the first time they've been on chemotherapy. Yeah. So once you get tunneled down through that, you get to a very small population, and it can be hard to find them. Patient's history can be quite complicated. You think maybe you found a patient, and then you interview them, and you find they've got some other condition or disease that is going to conflate getting statistics out of them in the end. That patient may not want to participate. Okay? We want as many people as possible, but we might have doctors that they don't want to participate in our trial, or they do want to participate in our trial, but they're not very good at returning results to the trial. They just want to get the drugs to give to their patients in the best possible way. Okay? And we also have competition for the same set of patients. Last year, we had a particular drug we rolled out to a set of cancer patients. We found that 6 months earlier, our rival pharmaceutical company had already basically ring-fenced that set of patients. They had a drug for exactly the same cohort. So how do we find these people? Now, there's an odd thing about a lot of these questions. You know, there are the formal questions and there are the questions in setting up the trials. And we started to hear them a lot. They're things like most basically, you know, how do we find more of this X type of patient in Spain or in France or Italy or wherever? Or we've set up a trial in Madrid and we don't want to expand out of that city. Can we find other people in that city? Okay, can we find other sites in that city? Or, you know, we've got so many practitioners on board, how can we get other practitioners in that hidden population? Okay? We'd like to work with X. They're good. We trust them. We know them. How can we find other people that they trust? There's this whole relationship thing there. They want to know about key figures. They want to know about influencers, opinion leaders, your keys and your calls, and all those other three-letter acronyms. And so there, in the hard data-driven world of pharma, we find ourselves confronted with a bunch of soft questions. Questions about social things, which we are notoriously bad at. Okay, so the solution. A bunch of us sitting around at AstraZeneca thinking about projects, and we said, why don't we build a social graph of trial practitioners? Facebook for oncology doctors. Okay, we can link these guys up by they've been participating in trials, they've published with each other. Okay, they work at the same institution, and we can ask questions about them. We can annotate that graph with various sources of data. And so we have this project Opsin, the Oncology Practitioner Social Interaction Network. And this is part of a larger AZ project, I might say, to streamline clinical trial practice. It's such a big of the drug process. We want to get everything working in a mechanical, reproducible way, have all the information at hand so they can be run as efficiently and as effectively as possible. Now, as it happens, all good ideas, of course, have been thought of beforehand. And it was somewhat my surprise that more than 10 years ago, in fact, in 2008, there were some people at AstraZeneca who did essentially the same thing. Hand-roll graph databases, scraping things off the web, pulling in data, and so forth, and managed to make themselves a sort of social graph from which they could find the key opinion leaders and things like that. So, okay, I'm not proud. I'm willing to do things better because we have much, much more information these days, much more what I'd call ambient information. Even those of us who like to eschew social media can't help but leave a digital trace all over the world. We have our LinkedIn profiles, we have publications, we have mentions of ourselves in the literature. We also have much better technical platforms for doing this, and there is a growing interest, as I said, in pharma in the whole graph area, particularly in knowledge graphs. That so far has largely been in biochemical and biological entities, but this could dovetail in nicely. Right? We also have this large body of internal data where people inside the company keep lists of the naughty and nice, who we like working with, who's been on one of our trials before, who's been really good at recruiting people. The downside, okay? Of course, a lot of our social graphs out there are done by the big hitters, your Facebooks and your Googles and so forth, who can afford to solve problems by just throwing big iron and big teams of people at them and big amounts of money. So we are starting from behind the eight ball on that. Also, if you go out there and you look at methodology and you try and find out, tell me who the most influential doctors are, tell me who the technicians are in this area and so forth, it is quite amazing to see how much of that, in fact, the bulk of that, is basically hand-curated lists, okay? Someone will give you a list of these are the guys. Where did that come from? Well, their opinion, basically. And that is not going to scale over 100,000 oncology practitioners. We can't do that, and we're interested in everyone, not just the top 1%. Also, relevant for us, the field is changing rapidly, and so relationships might change, drugs might change, therapies might change, and so forth. So how do we deal with that? We have these issues with data governance and privacy, which I've hinted at already, and which actually become a bit of a major problem for us. And while initially we said, let's go through social media and so forth, it's an open question as to whether a significant number of scientists actually use Twitter. So, data sources. As said, there's a lot of data out there. And as said, we have this high level of ambient information, okay? We have PubMed, the great repository of biomedical publications, and reflected in Google Scholar and Microsoft Academic, and that stuff is almost trivial to get into our system. We also have really neat things like clinicaltrials.gov, so that's every clinical trial run in the United States. And we've got efforts like Open Trials by Ben Goldacre, who's basically scraping the entire world's records of clinical trials, structuring it, and sticking it into a database himself, so it's searchable and examinable. Scientists loving to give meetings and awards to each other and so forth. We have another level of ambient information in there. We have all this internal data as well, and LinkedIn and so forth. Where we run into problems, oddly enough, is not our PubMed, it's not our LinkedIn, it's not our ClinTrials or anything like that. It's that association information. Now, many of our big organizations, our big hitters, will fly very close to the line, or in fact past it, on data rights and security. I mean, you would have heard recently about this facial recognition firm that basically just scraped images off Facebook and Instagram, using it to build a database. It's clearly unethical, beyond the line and so forth, but no one's going after them. We don't have that luxury. We have to do this strictly. So a lot of time and effort has been spent just trying to get like a membership list and lists of abstracts of practitioner organizations. So there is a warning to you all. In a way, the smaller the organization, the more difficult and protracted that procedure becomes. Designing the actual system, and this has two aspects in there, one being the actual platform, the other being the schema, and so forth. Now, as I said, several of our predecessors have already done something like this some years ago, but there was one thread of problems that continued through all of that, which was that none of those efforts were sustainable. They did up something in R. Someone banged out a Python script, okay? They hand-rolled their own graph DB. And so none of those were suitable for operationalization, and this has got to be. This has deliberately got to plug into other systems so they can query it. So this is where Grakn steps in. Of course, there are other graph systems out there, but knowing from previous experience back at the university and so forth, that we're very interested in edges, the provenance of edges, and making statements about edges. Really, we were talking about hypergraphs, and Grakn just makes that so much easier. Now, our entity selection and design. Being scientists, naturally, we love formal schema and ontologies, and the first thing we did was run off to say what prior art there was in there. And fortunately, Thomas was able to talk me down and say, really, just what questions are it that you want to ask? So we went for the dumbest thing possible to solve this. Okay? So we just have a graph of people, person with a capital P. We have a series of collaborations, and there will be things like clinical trials, grants, publications. There will be institutions that they worked at, okay? Each of those relationships has a temporal element, years in which it took place in, okay? Social media doesn't quite map to that model yet, and this is something we're struggling with. And we've made a couple of great simplifying assumptions. We have a data cut, the data we're using basically for the last 5 years. And this just makes things simple for us. If you worked 20 years ago with someone, maybe you've got a great relationship with them, maybe not. Maybe you've changed field, maybe not. If we take a 5-year window, we avoid all of those questions. We say basically everything is going to be the same. Okay? And we don't have to worry about the atropine. We don't have to worry about knowledge changing. 5 years is a fairly tight window. It also saves us from the bias of information towards the present. Right? So we needn't worry about the fact that in 2010 we had much less information than we do in the current day. And for certain questions of evolution, we just produce a different version of the graph for each year, a cumulative graph. Disambiguation is, of course, a big issue as well, too. I was fortunate enough when I was back in university to have a mentor, Paul Riley, his name was. A great man, but he had his stage name, his publication name was Paul F.E.B. Riley. I think F was Francis, E was Eustace, and B he gave to himself, so he could distinguish himself from every other Paul F.E. Riley. And in the literature, we have all these dozens of lookalikes in there floating around. How do we separate them out? How do we separate them out, indeed, given that we're going to be using lots of different data sources? Now, there are many, many disambiguation things out there, but again, we had 2 lucky breaks. One, we decided we only had to be mostly correct. Okay, we're happy with being 80% correct, right? That will get us close enough. Also, some of our predecessors inside AstraZeneca had already pre-digested PubMed, okay? Filtered through it, disambiguated the names, disambiguated the institutions, and done that for us. So that was our gold standard. Map against PubMed, and then go back out. So we circled around that problem. And what do we end up with? Well, as said, this is a work in progress, definitely, okay? But if there's one lesson there, it's that some of the hard questions are actually quite easy, and some of the seemingly easy questions are not so. So that question about we need poor practitioners, who do our guys know, is trivial to ask in the system. Trace through a collaboration to someone else, boom. It's easy to do, and you can rank that by the number of connections. You can rank that by how recent or temporal it is. You can rank that by, I want these people to have worked together in a trial before. I want these people to have worked in the building, or so forth. That becomes quite simple. So we can say that Dr. X knows Dr. Y because they were on this trial or they published together. That question, which was just so common, is just solved trivially. What's not so easy to solve is the question of location. So as said, we very often get this question about, okay, what other sites are there in the same city? Or, what other patients might there be in the same city so they can come to one of our sites, be referred from those hospitals? And this is a very simple thing to express, but actually becomes quite difficult to solve. Because what does nearby mean? What even does city mean? Because if you're out in rural France or whatever, you know, there may not actually be a city or so forth. So, and there's also the problem with institutes with multiple sites. Not so bad in Europe, terrible in the United States, where you have these large mega hospitals spread across the country. So one of our first attempts we're working through is to use these things called nut codes. Okay, nut codes is, I think it may have originated from the UK, where essentially it's a set of nomenclature for a series of units that is harmonized across the UK and US, where you talk about a NUTS1 unit will have around about 3 to 7 million people in it. It's a geographically defined area. Then you'll have a NUTS2, which will have around about 500,000 in it, and a NUTS3, which will go down to around about 100,000, so forth. And in Britain, that 3 million might mean like a county or something like that. If you go to Austria, it might be an aggregation of states or so forth. Okay? There is a harmony in there. That's still not particularly great, because you could be just across a boundary, of course, within a NUTS code, and we end up having to assign things to region, and we end up with these thousands of these regions sitting in our graph trying to sort things out. So at the moment, we're in transition of trying to just put a lat long on our institutes and work out a distance from there. Still not a great solution. If anyone has anything better, I would be so happy to hear. Finding communities and central figures in those communities is also another one of those incredibly trivial tasks. Okay? We just say, what are the clusters in your graph? Dead easy. Then you say, who are the key people in those graphs? Who are the ones who are the most central? Who are the ones who have the most connections in there? Now, unsurprisingly, that largely duplicates discipline boundaries, with a caveat that comes up in a moment. But we are looking at using the shifts in the centrality and connectivity across the years to try and track who is a rising star. Who do we want to try and recruit to our side, or would be good to talk to, because in 2 years' time, they're going to be a great figure. But clusters is one of my things. That's my petition. And what do you do with a hairball? Because almost anything is a hairball in there. Almost anything, any interesting data, is just connected to everything else, okay? Everything can eventually trace a path to any other point. Okay, you have many, many links in there of unknown value. Okay, how can we get down to an actual, how can we distill that to a core cluster in there? And if you chase the literature on this, I'll say that you can look for things like graph simplification, sparsification, shrinking, and so forth. But invariably, we gave it a go in another way. There is a quite intuitive method that, curiously enough, arose from biology, a weighted correlation network analysis. Now, the way that works is that you essentially transform your graph into a set of kind of similarities between nodes. Then you apply a threshold that slowly breaks apart the network, okay, as you make that threshold stricter and stricter. Now, here's the magic step in there. Incidentally, this is used for working out gene correlations and so forth. So you slowly break apart your network and see how it adheres to being a scale-free topology, the idea of a natural network. You know, you get these people like Strogatz or so forth who say that your small networks and things like that have a particular geometric shape to them, a natural shape. So there is a feeling that that is the right shape for a cluster to be. So while we veer towards that, we also try and keep the graph as connected as possible. So you simultaneously try to optimize for connectivity and for scale-free behavior, and you stop at that point and you say, that is an informative graph. And it's a quite nice, intuitive method that makes sense to me, so I like it. And here comes another part about me being a cluster geek, which I normally use to do things like patient types and so forth. How can we find types of patients or types of disease out in the world? How can we find types of behaviors of biological entities? And everyone of course does clustering in their first year of data science, right? It's just the simplest thing ever. But there is this body of literature out there almost about the philosophy of clustering. What is a cluster? Is a cluster meaningful? You can make clusters from random data, but how do we talk about whether a cluster is ontologically real? What is a cluster? Is it a thing rather than just an artifact? And if you use different methods of clustering on different sets of data, you will get vastly different answers out there. And we can apply this as a wider question to all of our social questions. How do we know that we've got the right answer? This is quite difficult. We can, of course, do things like direct validation of clusters. Do they make a good cluster? Okay. We can subsample to see that our answers are stable. We can use different types of data to see if they come up with the same clusters. Big hint, they don't, because people tend to have different sort of reactions. I don't actually know how to solve this problem, and it bothers me a little bit, because there have been solutions developed before where people say, this is the best person, this is the answer, and so forth, and there is no way of showing whether that is right or not. And while it is very tempting to settle merely for being useful, I would actually like to know to some extent whether I am correct. So, as said, we are a work in progress, and where are we going? And here it is. So there's a long idea in science and so forth about idea transmission. Even before we started to talk about memes, people said, how do scientific theories arise? How do they take over communities and so forth? And we can actually track this in this graph. We can look for certain keywords or concepts in clinical trials or in papers or so forth referring to a particular therapy or drug or result. Everyone who is attached to that publication or trial has that idea, and over the years we can watch that idea percolate through the community. And that's an interesting and fascinating thing. But why do we want to do that? Well, we want to see who transmits those ideas. Because not everyone is equal, okay? There are good doctors and there are bad doctors. Incidentally, this is the 6th book in the Dishy Doctor series. It went downhill after the 4th. So everyone in the trade knows there are people who they're good at leading opinion. There are people who are good at transmitting opinion. There are investigators who are good at leading trials. There are investigators who are actually good at doing the work on trials. Okay? We have people that we trust that deliver, so forth. How can we identify those different types of people? They might be equally well connected. They might be at similar points in the graph. How do we lead them out? And so this is us trying to identify what types of roles people play in the community. And so this is where social media starts to move in, and also things that are more opinionated, commentary, editorials, and so forth. We have to do social analysis on those, and therefore try and get some nuance on the opinion and ideas that are going through, and see if we can identify the type of people in our graphs there. So, even in the hard data-driven world of pharma, we still have a lot of soft question. It's a human enterprise. We're going to have human questions in there. And we have a lot of ambient data out there that we can use to tackle these. Data rights is not a question to skimp on. It is a difficult one. You need to get that right. Okay? Some of our hard things are easy. Some of our easy things are hard. But the whole idea of whether you're knowing whether you're right is a tricky one. And just finally, penultimately, the wonderful team here. Domingo had previously done Twitter analysis back in Imperial and was one of the people who originated this result here. Bill and Maya done great work in there, and Gabby is our grateful sponsor from within Astra who gave us a go on this project and provided funding. And of course, Grakn. Now, AstraZeneca and pharma in general, but really you want to be thinking about AstraZeneca, okay? We cannot get enough people, okay? We cannot get enough good people. Okay? We're just continually recruiting data scientists and things like that. We have some very sweet offices in central Cambridge, 5 minutes from the railway station. We have good coffee, and we don't make you work late. So there's an URL there. Look it up. Thank you.